just like in a library, you see, library is a very segregated place. There are thousands of books in library, but it is very easy to find which book you want to read, right? And why is that? Because everything is there is a classification system. Everything is segregated on the basis of what? So what is one thing, the first requirement to make classifications, to put something in classes? What do we need, Maha?
Characteristics.
Yes. So classification is done on the basis of some characteristics. Now, very early, when biologists started characterizing animals or living systems around, there was a person called Linnaeus. Carolus Linnaeus. Have you heard of this name?
Yes, sir.
Linnaeus came up with a two-kingdom classification, right? You must have studied this. So he said that everything can be classified, everything which is living can be classified into two kingdoms. Either they are plants, okay, or they are animals. Correct? But he was not correct, because in his time, either you could see plants around you or animals around you. But if you go to very detailed observation of the nature, do we only have plants and animals in our surroundings?
No.
So how will you define plants? Tell me first. Let's start there, Ma. How will you define a plant? According to a two-kingdom plant. Very good. So they are photosynthetic, which means they can synthesize their own food, with the help of chlorophyll. Chlorophyll is a green pigment that gives plant the green color, and it has the ability to chemically convert the light energy to trap the light energy and basically convert it into chemical energy, right? So they are photosynthetic. Can I also say that they are, what is the other word for photosynthetic?
Autotrophs.
Autotrophs, yes, perfect. So they are autotrophs. Now, remember I told you that I always believe in studying biology through terms. Do you know what does the word photo means, Maha?
No.
Photo means light. So have you made a copy?
Not yet.
No, for biology you have not made a notebook? Please make
Yes, I do have a notebook for biology.
You have a notebook? Okay, very good.
Yes.
So have you left few pages, like I said, either in the beginning or in the end? Okay, now go there, write down your first word. The word photo in science means light. You must have heard of a term called photon in physics. Right? Photon. Have you heard of photon?
Yes.
These are, what are photons? Photons are small energy packets of the light. Okay. Light is an energy, right? Light is a form of radiation. And the word synthesis or synthetic. So this word comes from synthesis. So they can do photosynthesis. Synthesis means to produce. Write down. Synthesis is to produce. Okay. So if an organism can produce its own food, its own nutrition in the presence of sunlight, by using sunlight, those organisms are called photosynthetic organisms, and they are plants. Similarly, the word auto, you know what does the word auto means in science?
No.
Auto means self. Have you heard of things called automatic? This machine is automatic.
Yes.
It means that it works on its own. You don't have to manually operate it all the time. Okay, those are automatic machines. Like if a washing machine is automatic, we call it where we just have to put the clothes, put down a program and run it. So it will wash the clothes, rinse the clothes, dry the clothes, and then give you a beep that everything is done. That's called an automatic machine, washing machine. Manual is where for every step you have to manually put things and start or twist some knob or something. You understand?
Yes, sir.
Yes. And those organisms which do photosynthesis are called autotrophic, because the word auto means, now you know,
Self.
self, and the word trophic means nutrition.
Oh.
So they derive their self nutrition. On the other hand, animals are heterotrophic. What do we mean by the word hetero? You have used this word hetero. So all these words are very, very important. Just keep writing it in your terminology section so that from next time you do not think biology is too much to remember. You just know it now.
Okay.
The word hetero, heterogeneous mixtures, homogeneous mixtures. The word trophic means nutrition here also, right? You now know, you
Yes.
will never forget trophic. What does the word hetero means? Heterotrophic mixture in chemistry? Yes, tell me. If you put, yes, ma, tell me.
No, I don't know.
So if you put sugar in water, it will be a homogeneous mixture, right? Where you cannot
Yes.
separate that there are two different things present. It will feel like one simple thing throughout. In a heterotrophic, heterogeneous mixture, for example, you can very easily distinguish that there are different subparts in that mixture, right? So the word hetero means different. Okay. So heterotrophic organisms rely on different sources for their nutrition. You understand?
Yes, sir.
Yes. And we rely on either plants or other animals to derive our nutrition. We cannot synthesize our nutrition in our own body. You know, if we could have done that, then we would not have evolved at all. You know that hunger is the biggest driver of evolution, right?
Yes.
Whatever we do for food. All the conquest in the world, you're like, why are you studying biology? Why do you want to become a doctor? Why do I want to become a researcher? Why are we doing science? You know, yes, you can say that, you know, I'm curious about it. I want to seek the knowledge. So there are two things. Either you want to seek the knowledge and help or make something for the future of humanity, make things in life better. But the major thing is so that you are employed, you are doing something, you are ensuring a steady flow of resources for yourself with which you can buy food and, you know, not be hungry. If
Yes.
our ancestors, plants are never hungry, right? That's why they never move. That's why they never fight wars. That's why they never, you know, conquer countries or make their own establishments or go out every morning to offices or read biology. Nothing. They are sorted. They just have to stand at one place and wait for the sun to rise. The sun will rise. They have nothing to do. So the sunlight will fall on their leaves. They have chlorophyll in their leaves. Those chlorophyll will trap the sunlight, the energy of that sunlight, convert it into chemical energy, and they will produce their own carbohydrate, starch in their own body, protein in their own body, and they will grow. No problem, like sorted life. I think many of us will like that kind of a life if given a
Yes.
chance. Yes. Are you also like one of them? Would you like to be a plant?
Yes.
Yes. Nice. Great. Yeah. Okay. Very good. So you understood first difference between plant and animalia is heterotrophic and autotrophic. That was okay. What else? What else? Both plant and animals are multicellular only, right?
Yes.
And eukaryotic. So what does the word eukaryotic means? I think we have discussed this. Eu means so when Linnaeus gave this system, he was not aware of eukaryotic and prokaryotic cells, that there can be a difference between the cell types of an organism. So plants, the word "you" everywhere in biology means true. You will hear this word multiple times. Eukaryotic, euchromatin, eubacteria. Everywhere it means true. True nucleus, true chromatin, true bacteria. Okay. So the word "you" means true, and karyotic comes from nucleus or nuclear arrangement. So eukaryotic are those cells which have a well-defined nucleus. Okay. So that is called a true nucleus. Yes or no?
Yes.
Now, in this true nucleus, there is a nuclear membrane present. Well-defined nucleus, so nuclear membrane is present. Nuclear membrane is present both in plants and in animals. But later on, we found that there is something called bacteria. And bacteria is a prokaryote. Now, can you tell me what does pro means?
Does not have a well-defined nucleus.
Yes, pro means primitive. Okay. And karyon means nucleus. So primitive nucleus. So this is, you are very well correct. It does not have a well-defined nucleus. Does not have well-defined nucleus, which means no nuclear membrane, right?
Yes.
So far, so good. You are understanding this?
Yes.
Now, if you just go through the two kingdom classification, and later on when bacteria were found, in which kingdom can we put bacteria? Can we put bacteria in Plantae or in Animalia?
None.
None. So clearly this two classification system, two kingdom classification was not enough, right, to accommodate all sorts of life. Then came a three kingdom classification because people started including bacteria, and then fungi were found. And then they realized, oh, a fungi is neither a bacteria, nor a plant, nor an animal. So they made a four kingdom classification. And then protozoans were found, like Plasmodium, which causes malaria. And then they were like, oh, this also does not fit in bacteria, fungi, plants, and animalia. So let's make a five kingdom classification. So the one that you have to study for your syllabus is a five kingdom classification. You know, five kingdom classification is also not able to accommodate all. There are something called viruses. You know that?
Yes.
Viruses fits neither in bacteria, nor in fungi, nor in protozoa, nor in plants, nor in animals. So viruses are very, very different from all other life forms because outside the host, they are not living at all. They are just like a non-living particle. Okay. They do not have a cytoplasm. But inside the host, they become active and start behaving like a living system, start doing transcription, translation, and starts infecting the host. So even five kingdom classification has misses some things which cannot be included, but you have to study five kingdom classification. So now let me tell you about five. So this is a major theme of this whole chapter 2. So today I will brief you about five kingdom classification. Then on Wednesday, we will go into the detail of it in the same half an hour thing. Okay. So
Okay.
I will ask you to go back and read the five kingdom classification and come back, and on Wednesday we will do it in detail. Now this five kingdom classification was given by, you know, who gave?
R.H. Whitaker.
Yes, Robert H. Whitaker. R.H. Whitaker. So he said, so as you told in the beginning, there has to be some characteristics, right? on the basis of which we make the classification. So let's see what were the characteristics he used. And then on the basis of it, there were 5 kingdoms. So let's first come to the class characteristic. First characteristic he used was cell type, whether it is a prokaryote or a eukaryote, because the first problem that came in a 2-kingdom classification was that both of them are eukaryotic, and then you figured out something is prokaryotic and you had to include another kingdom. So he said, let's talk about cell type. So starting with the first kingdom, which is Monera, which contains all the bacteria. Monera are prokaryotic. Okay? Clear?
Yes.
Then the second one is Protista. Protista is eukaryotic. So from Protista, eukaryotes start. This is the simplest eukaryotic. And then we have fungi. When we will finish this table, you will also be able to distinguish between all. Fungi is also eukaryotic. Then we have Plantae, again eukaryotic, and Animalia, of course eukaryotic. Okay. The second characteristic he chose was cell wall. Is there a cell wall present or not? So cell wall is an additional layer over the cell membrane. You know cell membrane?
Yes.
Yeah. Okay. So cell wall is present in Monera. So I'm writing it's present. Okay. And it is made up of polysaccharide plus peptidoglycan. Okay. So it is present. In Protista, it is present in some, not all. So this is one difference, okay? And of course it is eukaryotic, so that is another difference. Then in fungi, it is present. It is present in all, and it is made up of chitin, a different type of cell wall. Then in Plantae also it is present, and it is made up of cellulose. That is how plants are very, very sturdy. And in Animalia, it is absent. We do not have a cell wall. Understood?
Yes.
Okay. Then he came to the third characteristic, which is what about the nuclear membrane? What about the nucleus? How is the nucleus? So if it is prokaryotic, if Monera is prokaryotic, the nuclear membrane will be absent. Okay. And it will be present in rest 4. Clear?
Yes.
Yeah. Then we go to some more advanced features like body organization. Body organization. Now, body organization is how is the body organized? So, Monera is unicellular. What is it?
Unicellular.
So, the body, oh, just one second. Hello, Iram. How are you?
Hello, Zoom. Yeah, I'm good. How are you?
Yeah, I'm good. So, you have a new batchmate, Maha. You can say hi to Maha. I'm just giving a brief recap of chapter 2 for Maha because that's the chapter that's going on in her school. And in some time I'll be starting with cell division. Okay, so you can either stay or just be there, and we'll wait for Aisha to join, and then we'll start. Okay,
Okay.
in that time I'm discussing this thing with Maha. You can also participate. Ultimately we have to do it in detail, but it's good if you will participate. Okay, so for Monera, which is basically the bacteria and everything, the body organization is unicellular. So they are just single-celled organism. Okay. Protista can actually be multicellular, but their body organizations remain cellular, which means they are a bunch of cells present together, but they do not behave like one unit. They behave like one unit, but each cell takes care of its own requirements. Do you understand, Maha? So for
Yes.
Monera, it is one single cell, no option. For Protista, it is like a bunch of cells together. So it will give you an appearance of being multicellular, but it is not multicellular. In reality, it is just a group of cells living together, but every cell is taking care of itself. From fungi onwards, we get a true multicellular organization. And here it is loose tissues. So fungi form loose tissues for the first time. Then Plantae, of course, it is multicellular. And it is more advanced. It has reached till tissue and organ. And animals being the most advanced or evolved, they're of course multicellular. This is V, and we have reached till organ system. So we have tissues, we have organs, and we have also have organ system. For example, cardiomyocytes. Remember, Maha, from the first introductory class? Cardiomyocytes are heart muscle cells.
Yes.
Yes. So those are cells. Then they will go on to form tissues together, heart tissue, which beats, right? Then all that tissue will make an organ, which is heart. And then heart, along with the blood vessels and the blood present in it, will form the circulatory system. So that is called the organ system. Do you understand?
Yes.
Very well. Okay, now the fifth thing, and the final thing, is the mode of nutrition on which it is classified. Now, so the mode of nutrition can, when Linnaeus gave the classification system, two kingdom classification, there was two modes, autotrophic, heterotrophic. But bacteria can be multiple. Bacteria can either be autotrophic, which means there are some photosynthetic bacteria. And in the autotrophic, they can be chemosynthetic. What does the word chemo means?
It uses chemicals.
Exactly. So bacteria can directly take chemicals from the environment. For example, if you need, Maha, let's say a human has a calcium deficiency. So should the human start eating chalk?
No.
Then should we start eating the shell of the egg because it is made up of calcium carbonate? Can we directly take chemicals from our surroundings and eat it?
No.
No. We have to depend on, we have to eat something, food, and then from there we have to absorb that chemical. But if bacteria need something like sulfur, phosphorus, chemical, they can just directly absorb it from the environment. That is called chemosynthetic. Hello, Aisha. Am I audible, Aisha?
Yes, sir.
Yes, so just give me 2-3 minutes. I'm just discussing something with Maha. She is your batchmate, new batchmate. She'll be joining us from today. Okay. Both Hiram and Aisha can say hi to Maha in your own canonical styles through using emojis. So one thing, Maha, you should know about Hiram and Aisha as your batchmates. I'll also introduce you all. They use emojis a lot. See, Aisha came up with something. What is this? Celebration? She is celebrating your presence in the batch.
Emoji.
Okay, that was okay. I need to learn a lot about it. Welcome emoji. Okay, anyway, so and what is this, Hiram? She's happy, I think. Okay, Ma, let's finish this discussion. Okay, so prokaryotic, which is bacteria, can either be autotrophic through chemosynthetic mode or photosynthetic mode. So there are some bacteria that can actually photosynthesize also. They have chlorophyll in them. Is it clear? Bacteria can also be heterotrophic. Okay, everyone, I think all of you should listen to this. Bacteria can either be autotrophic and they can also be heterotrophic. If a bacteria is heterotrophic, then either it is saprophytic. You know what does the word sapro means? Sapro.
No.
Sapro means feeding the dead and decaying. Write it down. The word sapro, saprophagy or saprophytic. Sapro means feeding the dead and decaying. They are saprophytes. And bacteria can be parasitic. What is parasites? Maha?
Feeding on living animals.
Yeah, but predators
Like
can
living
also
on
feed
them.
on by living either on them or inside
them,
them. Yeah?
Yeah.
If they're living on them, it is called ectoparasite. Ecto means outside. Okay. If they're living inside the body, they are called endoparasite, inside. Okay.
Yeah.
Yeah. Okay. So this is the mode of nutrition, and bacteria can have all different sorts of mode of nutrition. If we come to Protista, Protista can also be autotrophic. And if they are autotrophic, they are mainly autotrophic through photosynthesis. They cannot do chemosynthesis. In fact, you can know, every one of you should pay attention here. Chemosynthetic is a characteristic feature of Monera only. Only bacteria can directly take up chemical. Any other autotrophic is photosynthetic. And they can also be heterotrophic. And if they are heterotrophic, they are through feeding, like holozoic. Okay. For example, catching the prey or feeding on prey. You all have read about amoeba? Hiram,
Yes.
Aisha, yeah, amoeba. How does amoeba catches the prey? Puts out the pseudopodia and engulfs the prey. Now coming to fungi, the mode of nutrition in fungi is only heterotrophic. It is not autotrophic. It is only heterotrophic. So this is what differentiates fungi from others. It is only heterotrophic, and in that also it is like bacteria. Either it is saprophytic or it is parasitic. So this is only heterotrophic. Plants are only autotrophic, right? And in this, there is only one mode for plants, which is photosynthetic. They do photosynthesis. There are some exceptions in this case, which we will talk about when we will do this chapter in detail. Or I will discuss it with Maha separately. And in animals, animals are only heterotrophic. And animals also feed on other live animals, but that is called predation. Holozoic. The word holo means complete. So we eat complete food, the full food, and then we digest it in our body. So holozoic organisms have a digestive system, because if you are eating everything complete, you have to digest it in the body. Or the other mode is saprophytic. You depend on dead and decaying matter, because when something dies, it already degrades. We do not have to digest it. So it starts rotting. Okay, so many animals feed on dead and decaying matter. So they do not need to depend on a very good digestive system. Everything is digested already in this saprophytic juice. So is this clear, Ma? The 5 kingdom classification? Have you noted
Yes,
this?
sir. Yes.
Noted everything? Okay, very well. So we'll continue this discussion next time. Okay. So now let's start with, you have your other batchmates have joined, Iram and Aisha. Okay, so let's start with, you can say hi to them. Iram, Aisha, are you there? Come back. We are starting. Yes, Iram.
Yes.
Yes. So we have Ma with us. She'll be joining us from today in this batch. Ma, you can just say hello, hi to Iram and Aisha.
Hi.
Hi.
See, there's a waving hand emoji. So you're going to get a lot of this. Okay, where were we, Iram? So today we'll be starting, just one moment, cell division. Okay, the chapter cell division. So Ma, you have an idea about cell division as a chapter?
Yeah, kind of.
Okay, so let's, just one moment, let me share the screen. Okay, cell division. Okay, everyone, so let's begin. So tell me, what do you understand by cell division, cell cycle and cell division? Iram, Aisha, Ma. So Iram and Aisha has already studied cell with me, but I am expecting, Maha, that you must be knowing the basics of the cell, right, from your junior classes. You know what is a cell, what are the components present in the cell, the nucleus, mitochondria, things like that, right? And you also know that cell divides. Yes?
Yes.
And this division happens because of it has a cell cycle. So cell has a cycle through which it divides. Now, first, let's start with cell cycle. Why do we call it a cycle? What is cell cycle?
Because it repeats.
Because it repeats. Good. So there are two kind of processes in biology. Either they are cyclic processes, or they are, if something is repeating, it is called a cycle. If something is non-repeating, it has a start and an end, then it is called a process. Now, in a process, if the end product comes back to the initial product, that becomes a cycle. So for example, glycolysis is a process, it's a pathway, but Krebs is a cycle, Krebs cycle. Okay. And if you don't know about them, no problem, we'll study about it. For example, water cycle, you all must have studied water cycle in your junior classes, I reckon, in environmental studies or something. Water from the oceans and the rivers evaporates and goes back in the clouds, and then the same water comes down. So we have limited water on the planet. It is the same water which keeps circulating. Yes? Yes or no? Yeah. So technically, a water never, the molecule of water stays pure. So when you wash something with water, the same water that is being used by us goes to the sewer systems. Through there, it gets to the treatment plants, gets treated, and then the treated water is thrown back into the rivers or is used to purify. The same water is again purified and is made fit to use again. So the same water we use and we think is, contaminated, is purified and sent back to us because we have limited water. So it becomes a water cycle. Same is with carbon cycle, nitrogen cycle, etc. So now we're coming to cell cycle. Now, first, why do you think that for a cell division is important?
To produce.
To reproduce, right? Yes, right. But what about organisms like human? We are multicellular organisms. So in us, do you think that cells are only dividing for the sake of reproduction? Or is there something else?
Growth.
Yes, one is growth. Very good. Growth, reproduction, two things I got. Any other thing you can think of where cell division is important? Any other biological process? Yes, Aisha. Sorry?
Respiration.
Respiration. How respiration, Aisha? What is your thought process behind saying respiration? Do cells have to divide to respire? A bacteria, when it is not dividing, when it is a unicellular organism, then also it is doing respiration, no?
Yes.
Yes, yes, yeah, right. So not respiration, because if you give me some logic that I cannot counter argue, I'll accept respiration. But right now, back to back to state that I cannot accept respiration. Okay, I'm so sorry, Aisha. Yeah, she's okay. She's cool with it. What else? For just simply growth and reproduction, nothing else. People, come on. What happens when you? How many of you learned how to ride bicycle? All three of you can ride bicycle?
So repair.
Exactly. And you must have fallen from that bicycle learning it, right? And you must have gotten bruise or a cut in your body. So what happens when we get injury? When we get an injury in the body, cells from that region either die or are cut off, right? Then they have to heal back. And for that, we need cell division so that we can fill up for the missing cells. So repair, wear and tear, very important. And anything else? Replacement. Do you think that you have the body with which you were born? No, right? You know, we keep cells in our body keeps dying. Every cell has an age. We as an organism grow and live for around 70-80 years. But each and every cell in our body does not live for 70-80 years, except for a very few, like heart cells or our brain cells, neurons. They live that long. But most of other cells, they keeps dying, and keep other cells keep replacing those cells, and that also happens through cell division. You understand? So growth, reproduction, wear and tear, and replacement, all these processes in our body which are happening through cell division. Is it clear, everyone? Yes or no?
Yes, sir.
So first, let's define cell cycle. Write down. The series of events, cell cycle is the series of events by which a cell replicates its genome, which is DNA. So we make the copy of the DNA, comma, synthesize other components, like mitochondria, organelles, ribosomes, etc., etc. And eventually divides to give rise to two daughter cells. Two daughter cells. There's no concept of a son cell in biology. The products of one cell when it divides, it's always called two daughter cells. Okay. Two daughter cells is known as cell cycle. Or is called the cell cycle. Clear? The definition? Now, this cell cycle has various different phases. So there are phases of the cell cycle in which the most, what you can say, longest phase is called the resting phase. So cell is mostly growing for the longest time in its life. It grows, and then when it reaches a level where it needs to divide, the division phase is very small as compared to the growth phase. Okay, so if I so write down phases. Let's come to the phases. Now we will discuss many things in phases of cell cycle. So basically, you can divide it into two phases. When it is dividing and when it is not dividing. So the first phase is called interphase. What does the word inter means in biology? I think Iram and Aisha will answer this. Intercellular transportation, intracellular, we just went through this whole thing over and over again. Between. Exactly. So Maha, you can write down the word inter in means between, and the word intra means, what does it means, Iram?
Within.
Within. Yes, great. So intra means within and inter means between. So interphase means between two phases, right? Okay, now these two phases are division phases. So interphase is also known as resting phase. When it is not dividing, it is resting. So we always rest between two things, right? Between two activities, you take a rest. Second is called M phase. M phase. Okay. The word M stands for mitosis. It is a cell division type. It can also stand for meiosis, but for the sake of simplicity, we will first discuss mitosis. Do you know about mitosis and meiosis also, everyone? If not, then we can spend some time there. Tell me, are you clear about mitosis and meiosis? Maha, Iram, Aisha, yes, no, something as an answer will be good.
Yes, sir.
Yes. So mitosis is called equational division. Okay. In mitosis, if a cell has, let's say, 10 kgs substance in it, then it will replicate everything to make 20 kgs and then will divide 10 and 10 equally. So that's called equational division. In meiosis, it is called reductional. If a cell has 10 kgs thing, it will divide it into 5 and 5 without replicating it. You understand, everyone?
Yes, sir.
Yes. This M phase is also known as the division phase. So broadly, a cell cycle can be divided into a resting phase and a division phase. Okay. Now, this cell cycle depends on organism to organism. the timing. So the duration, write down the duration of cell cycle varies among organisms. Some you have to remember. Bacteria, for example, E. coli. You know, one cell cycle of E. coli is around 20 minutes. So it divides in 20 minutes, completes the cell cycle in 20 minutes. For yeast, you know what is a yeast? Yeast, baker's yeast, it's a fungi. Okay, it is the only unicellular fungi. It is used for baking the cakes. You put baker's yeast while baking the cake. Anyone of you have baked cake?
Yes.
Yes. So do you use something called baking
Yes, sir.
yeast in that? Yeast has a 90-minute long cell cycle. And if you talk about a human cell, okay, mostly human cells do it on the scale of hours. So it is around 24 hours, one day in some cases. So do you see that it is very, very different? And most of it is because of this resting phase, interphase. So how long is the interphase? It decides how long will be the whole cell cycle, because cell cycle has two things, resting and division time. Now let's write down something about it. Write down. The interphase, also known as the resting phase, the interphase, also known as the resting phase, is or covers around 95%, around 95% of the duration of cell cycle. When I first read this, I started realizing all the cells in my body are pretty much like me. When I think of doing something, now this habit has changed over time a little bit, but I think from 95% it has come to 80 or 70. But whenever we feels like, okay, today I have to do this, we spend a lot of time just thinking, or just resting. Okay, I'll just do it. It's called procrastination. Are you aware of this? Maha just, like very early in today's class when we're discussing, Maha told me that if she was given an option to live like a tree, she will take it. Right, Maha? She will happily become a plant and just do nothing. Bask in the sun. Enjoy the cold breeze, the rain. And if there is pollution, she will convert that pollution into starch. She will just live a producer's life. Happy, no daily routine, nothing. So our cells are just like us. They spend around 95% time just resting and growing. They are eating and growing. And when they decide to divide, they just do it in 5% of the time, very fast. Why are you sad? Are you very hardworking, very active person? Very opposite of this. Are you not happy with how cells behave?
No, sir.
Are you happy? Or can you just relate? Oh, this is me. This is not cells.
Yeah.
Yeah, because we all can actually. So whenever you think about cells from now onwards, just remember, and when I teach students cell division, I tell them that cell division is a program. It's an event. And when I will start teaching you about it, I very closely try to, like in parallel, try to give the analogy of a wedding, a marriage. What is a marriage? Tell me. You must have attended weddings and marriages. What is it? What happens in a marriage? In any marriage or wedding? Sorry, two people get married, right? It's the union of two people. And if I ask you, the most crucial part of the marriage, of the wedding, what is that most crucial part? Whatever culture it is, wedding just lasts for the actual crucial part, just lasts for not more than 10 minutes. Yes. Either you have to agree to it 3 times, or you have to just say I do and you may kiss the bride, or you have to just go around the fire and put the Mangal Sutra. That is the most crucial part, right? Yes or no? But the whole event of wedding, how long that spans? Months, right?
Yes.
Hours, months. Family starts coming and all these things starts happening, some this rasam and that rasam and all that thing, mehndi, haldi, all. I think like people with time are fine, they have more and more time, so they are adding on to the menu of these things. Like personally believe, because when my parents got married, I asked them what happened. They're like, we got married. When now I go and attend some marriages, it's like a whole list of things that will happen from day 1 to day 6. Then on day 7th will be the actual marriage. So that's how you can very easily understand a cell cycle. So the most important event in a cell division cycle is when all the chromosomes are together present on the metaphase plate. Then spindles come from one side, spindle comes from other side, take half of the genome in one direction, other half in another direction, and the cell divides. Yes, done. But before that, lots and lots of preparation, prophase, metaphase, growth phase, G0, G1, G2, all of this happens. So this is the analogy, okay? So for next few classes, come with a vibe of you are listening to a wedding story here. The wedding story here is intracellular wedding story. Okay, Maisha and Maha.
Yes.
So the interphase is 95% of the duration. M phase is only 5%. Now write down. The M phase represents the phase. M phase represents you can write in bracket 5%. M phase represents the phase when the actual cell division occurs. And this is the one that we have to study in detail. So feel happy that you only have to go into the details of the 5%, because the rest 95% does not have any details as such. It's just what you and me do on a Sunday. Yeah. So the phase when the actual cell division happens. Yes, Aisha. This is the only batch that I teach where I come prepared that today I'm going to learn at least 2 new emojis that I can send in my WhatsApp. And I've actually learned from Iran Maisha. So I want to thank both of you. Apart from a very few like smiling emojis or this, I did not use much. But now I realize that yes, this one, and the celebration one, I've started using that. Okay, now let's come to M phase. So write down, where do M phase? So first we will go into the details of M phase. Okay, write down M phase. Oh, okay. Before that, let me give you a brief outlook of all the phases using a pie chart. So let's say, let's make a, let's say this is where the cell this. At this point, the cell starts to divide. Okay, so this is where M phase is starting. So it will just go till here. Okay, this is the starting. Let me first make it, then you copy. Okay, starting of M phase or the cell division, and it's going in this direction. So in between this, we will have prophase, we will have metaphase, we will have anaphase, we will have telophase, and you will have cytokinesis. So I'm marking them as A, B, C, D, E, and I'm writing it here. A is prophase, B is metaphase, C is anaphase, D is telophase. Just one moment. Yeah. Telophase, and E is the cytokinesis. So from A to D is your M phase. Okay, everyone? After the M phase, there are two things that can happen. So the cell goes into a very long stage, which is called G1, growth phase 1, G1, which has a S phase, which is called synthesis phase. And then it has a G2, growth phase 2. Is it clear? So you realize that only this part from here to here is actually that we have to study in detail. This is the M phase. Rest everything, if I use green, so from here till here, it is called interphase. Draw this diagram. Okay, before that, one more thing. Let me use some other color. Let me use a red color. Sometimes after the cell division, okay, the cell, instead of going into G1, S or G2, it just goes into a mode which is called G0 or G0. Now this G0 phase happens in very few kind of cells, and it is called quiescent phase. Quiescent phase means the cell does not grow or divide in this phase. Okay, no cell growth or division. Do you understand? So G0 is like kind of, it's stuck. The cell cannot participate in further cell cycles. You understand, Maha, Iram, Aisha?
Yes,
it makes
sir.
sense? Yes, Maha, please.
No, so me. So once
Yeah,
if the
Iram.
cell enters the G0 phase, will the cell be able to come back to G1 phase?
It may, depending on the circumstances. I will. So G0 is a specialized. All cells do not go into G0 phase. For example, your neurons. Our neurons are in G0 phase. After once they are formed, they never divide again. They do not divide and form more neurons. If a neuron has to be formed in the brain, it has to be formed from the stem cells, neural stem cells, but a neuron cannot divide further because it is out of this whole cycle thing. It is like when you say, I'm out of this business. I no longer want to participate in this kind of a cycle. So to come out of the cycle and get into a arrested state is called G0 phase. So this is a arrested state with respect to cell cycle. It is called cell cycle arrest. Right down.
Okay. So what will the cell do in G0 phase?
It will just be and let be. What do we do on Sundays? That's all. It will Netflix and chill. Cool. Whatever it is it functions like neurons. What they do whatever they do every day. They do the job. They make you feel things. You can process things, but then they are just out of the cell cycle. It's not like they are dead. They are living. They are carrying their functions. They are doing everything that they are supposed to do, but they just do not grow any further. They do not go into the synthesis phase, replicate their DNA, then go into the prophase, metaphase, anaphase, telophase. No, they don't do this business anymore. Okay. So it's called cell cycle arrest. That's all. No, they do not participate in the cell cycle.
Sir,
Yes, Ma.
what does the cell do in the interphase?
Sorry?
What does the cell do in the interphase?
Yes, in the interphase, it does a lot of different things. So first it grows. So after the division, when you reach E is cytokinesis. Now tell me, Maha, what does the word cyto means?
Cell.
Very good. And what does the word kinesis means? We did it. Iram?
I forgot.
Iram, time to be a teacher. Come on.
Yes.
What does the word kinesis means? Kinesis.
Division.
Iram is like
division.
Iram is like, which one? You just tell me which one. Good. So cyto means cell and kinesis means division. So it is cell division. So if a bigger cell is dividing to form two cells, it is very, very logical and it is intuitional to think that both the two daughter cells will be smaller in size, at least half the size of the mother cell. Yes, Maha?
Yes.
So first it grows. So G1 is called growth 1. G1 stands for growth phase 1. Okay. Then S. S is for synthesis phase. So first it grows in the interphase, then it reaches some size, then it synthesizes many things which are required now because the cell is bigger, so you need more things, more proteins, right? Then again G2, in which some additional growth is required if the cell is, so G2 will come if the cell decides to divide further. Again, if the cell decides to go into another cell cycle, then it will again grow a little, go into the cell cycle, then divide and become smaller. You understand? Growth phase 1, growth phase 2, and S phase. So this is the interphase.
So,
yes, Hiram.
So you said that neurons do not divide, right? So
Yes.
They are fixed in number?
Yes. The neurons with which you are born, they never divide. They are fixed. But latest research tells us that we used to believe, it's not because I'm doing my specialization in neuroscience now, continuing my research in neuroscience. I can tell you that no, there's no O in that. It's just one field of biology. Okay. So I'm interested in neuroscience. So this is a question I can just go on for lengths discussing, but because it's my favorite area. So neurons with which we are born, do you know that when a human baby is born, the brain is not developed properly? It is born with a half-baked brain. That's why babies just after the birth do not know how to balance themselves, do not know how any motor skills. They cannot walk, they cannot talk, they cannot, they do not have emotions like fear of something. If they will see probably something which is harmful, they will just move towards it. They do not know that this is harmful. They do not know that I have to, get away from this thing. You understand? Yes or no?
Yes.
A human baby need a lot of protection. because we are born with a plastic brain. Plastic means not that the plastic that we see in the real world, the problematic plastic. No, plastic means which is still not fixed. We can mold it. Okay, we can train it, and still new neurons are being formed, new connections are being formed for some time. But after the birth, these neurons do not divide. They form more and more connections with each other. So if you know how a neuron looks like, it has a body. You must be knowing about it. It has a cell body. It has dendrites, right? And it has a very long axon, correct? And this axon can form 1, 2, 3, 4, multiple connections with one neuron, two neuron, three other neurons. So these connections are formed. The more number of connections you have in your brain, the more multitasking you can be, or the more amount of facts you can remember or skills you can learn. So it is just like a forest. Our brain is like a forest. In a forest, the path which is the most traveled. So let's say there is a small road, not road, what should we call it? In Hindi, we call it pagdandi. That area where if you walk on grass, if multiple people walk on the grass, you see that there is a way. Are you people understanding?
Yes, sir.
There should be a good word for it in English. Let me figure out. How many is anyone of you understands Hindi?
Yes, sir.
Yes. So what do we call pagdandi in Hindi? Sorry, pagdandi in English. Track or a pavement. Yes, a kind of a sidewalk or a track that gets created in a forest. So if say multiple peoples are using the same track of the forest area every day, then you will see that because of constantly walking on the grass in that region will die, right? And there will be a space created between the grass. So basically that track will be strengthened. The more it is walked by, the more it gets strengthened. And the others, if you stop using that again, then grasses will take over again. You understand? If you go after 2-3 years, then that track will be covered. You will not be able to find the track easily. So our brain is just like that. The more you use connections, let's say some neurons are making connections where you are learning how to play a guitar. So when you are learning that motor skill, you are making more connections in your brain. And the more you practice it, the more those connections get strengthened. If you are practicing, if you become a part of a debating society, or you are doing something artistic, you are a painter, writer, or you are studying science, so things get strengthened. If you like mathematics, then your mathematical ability, the neurons which are playing a part in your computational abilities get strengthened. They make more and more connections. So you become better and better with calculations. Okay. So whatever you use, your abilities, those neurons get strengthened. Those forest patches gets marked. Whatever you don't use, it gets weakened, and over time loses connections. So that's how brain works. So after the birth, you're right, that the neurons with which we are born, they do not divide, but new neurons can be formed in the brain. This is the latest research suggests and tells us, it's proven that we can make new neurons to compensate for the lost neurons in our brain. And if we can understand this thing very well, we can, in near future, use it as a therapy to treat many neurodegenerative diseases like Alzheimer's, dementia, etc. Dementia, when people get old, they tend to forget a lot. They even forget their own house, they forget their addresses. But some things they don't forget. Like if you know how to drive a car, you will not forget how to drive a car very easily. Something are motor skills. If you know how to swim, it is a motor skill. But some things like your address, your bank account, your phone number, your kid's name, you will forget that. That's called dementia. So that also happens because neurons starts dying, and we cannot divide neurons to increase their number. So we have to start neurogenesis from the beginning. That can be done. But yes, neurons don't divide. Okay, Iram, does that answer your questions?
Okay. Yeah.
Okay. So this which goes in the G0 phase no longer participate in cell cycle or no longer undergo cell cycle. You can give an example of cells in G0 phase is our neurons.
Sir, cardiac muscles also don't divide, right?
Yes, cardiac
cells.
muscles also don't divide. Yes. So if we have a heart injury, but you know, it is true for humans. When we are in embryonic stage, our cardiac muscles can regenerate. But when we are born, then our heart cells do not regenerate. You know, they cannot divide and form new cells. That's why any heart injury remains there as a scar forever. But there are many organisms that can regenerate their heart also. For example, zebrafish. It's a small fish that can make the heart regenerate. It can divide the heart cells, but humans cannot. Yes, you're right. Okay, shall I move forward now? So clearly, this thing that we are going to study in this chapter is not very, very simple, but I'll try to simplify it for your syllabus. But cell cycle is one thing which the more we know about it, the less we think we know. You know, there is more to find. Okay, now let's come to M phase. Write down. The M phase starts. M phase. The M phase starts with karyokinesis. Now, Maha will tell me what is karyokinesis.
No, I forgot what that is.
Okay, you tell me what is kinesis.
Yeah, division.
Very good. And now Aisha will tell me what is karyo. Eukaryotes. Yes, Aisha. Very good. It means nucleus. So what does karyokinesis means?
Division of.
Yes, nuclear division. Right? So it's simple. If cell has to divide, then it first have to divide the nucleus which is inside. So when nucleus divides, the nuclear membrane is gone. So first is, write down, it starts with nuclear M phase starts with karyokinesis. In bracket, write down nuclear division. In which, so in karyokinesis, daughter chromosomes are separated. Okay. So chromosomes also duplicate in copies and become double. Then half goes on one side, half goes on other side. That's called karyokinesis. So it starts with karyokinesis, second point, and is followed by cytokinesis. So M phase starts with karyokinesis, followed by cytokinesis. This you already know, right? Now, M phase is clear, just the broader outlook is clear.
Yes, sir.
Now, something about the interphase. Write down some interesting things about interphase that have been asked in entrances. Interphase. This is where I will answer. I think Iram asked the question about interphase, right? Who asked, what does a cell do in interphase?
I did.
Okay, Ma asked. Okay, fine. So in interphase, write down, though it is commonly known as resting phase, though it is commonly called as resting phase, you know, the cell is not exactly resting. It's not like it's not doing anything. That is true for us also. When we are resting, it's not like we are not doing anything. No one rests. You can sleep. Even when you are sleeping, it's not like you are not doing anything. As an organism, you are doing a lot of things when you are sleeping. You are healing the system. So that's why they said that if someone is ill, if someone has an infection, they should sleep, right? They should take the medicines and sleep. Why is sleep advised in all sorts of illnesses? Can anyone tell me? What does sleep do? It brings us to a state where our brain can focus on involuntary functions like dealing with the infection, killing. Because when you are awake, when you are not sleeping, you are also focusing on voluntary things. Like if you are moving your hand, if you are looking at something, then you are processing what you are seeing. All these things are happening. You may be eating something, watching, hearing, talking. So when you sleep, you cut down all those voluntary actions and body can focus more on healing, repair, growth now. So similarly, during interphase, it is called resting phase, but the cell is actually preparing. Write down. During this phase, the cell prepares for the upcoming cell division. Cell division by undergoing both cell growth and DNA replication. So both of these two things happens in interphase. First thing that happens is cell growth. Okay. And then DNA replication. Now this growth phase is also in some books is written as gap phase. Gap 1, and this is called gap 2. But just remember that the growth is an integral part of, even if it is gap phase, growth is happening. Now this interphase can be divided into 3 parts. First is G1 phase, which I told you it can be either known as growth or gap 1. And second is S phase, which is synthesis. And the third is G2. I'm not including G0 here because I told you it is an exceptional condition where only few cells get into G0. And if they get into G0, they will not get into G1, S or G2. Okay, clear, all 3 of you?
Yes, sir.
Yeah. Now write down about G1 first. G1. So first know that in S phase, okay, DNA replication happens, because DNA replication is basically synthesis of DNA, right? When you are replicating something, you are making new. Yes or no? So DNA replication happens in S phase. So write down what happens in G1 phase. G1 phase is the interval between G1 phase is the interval between mitosis and DNA replication. So before G1, if you look at this thing here, see, Before G1, mitosis were happening, right? M phase was there. Yes or no? And after
Yes.
G1, there will be DNA synthesis or all sorts of synthesis, protein, DNA, everything. So G1 is between mitosis and DNA replication. And this is a question. It is a neat question. Which of the following phases lies in between cytokinesis and DNA replication? It is G1 because E here is cytokinesis. Remember? So write down G1 is between mitosis or M phase or cytokinesis and DNA replication. Now what happens? Write down during the G1 phase. During the G1 phase, the cell is metabolically active. Okay. In a G0 phase, the cell does not grow. Okay. So though it may be doing some metabolic activities, but it is not growing in size. In G1 phase, it is metabolically active and continuously grow in size. In G1 phase, the cell is metabolically active and continuously grow in size, but do not replicate the DNA. Is G1 phase clear? All three of you?
Yes, sir.
Yes. Now come to the S phase, write down. S phase or synthesis phase during S phase, the amount of DNA in the cell doubles. During S phase, the amount of DNA in the cell doubles. Okay, but next point, but there is no increase in the chromosome number. There is no increase in the chromosome number. Now, tell me, how is it happening then? If you are increasing the DNA content but not increasing the number of chromosome, how is it happening? Just think about it. I'll be just coming in 1 minute. Okay, Maha, Iram, just need a break. 1 minute. Okay, just think, how can we double up the DNA content without increasing the chromosome number? I'll be back in 1 minute. Yes, I'm back, girls. Are you there?
Yes, sir.
Hiram? Yes. Tell me, how do you think the DNA amount can get double, but the number of chromosomes still are the same?
By replicating it,
By replicating what?
the DNA.
But if you replicate the DNA is packed in the form of chromosome, no?
Yes, but it will be the same amount of chromosomes.
So let me.
Sir,
Yes,
I think they maintain the same ploidy. That's why.
exactly. They have to maintain the same ploidy. Perfect. And what do we mean by ploidy? The content of the
N.
cell, n number of the cell. Okay, I'll come to ploidy later, but tell me, have you seen what is the, when I say chromosome, do you have this picture in your head? Roughly? Is this the picture which you have in your head when I talk about chromosome?
Yes.
Yes. So this is the answer. This is already 2 times the DNA because this is one chromatid and this is the second chromatid. And both of them in this form are called sister chromatids. What are they called? So this is a chromosome in which these two are sister chromatids. They are identical. Chromosome, actually, we should know that it is like this. This is a chromosome. Okay. When S phase comes, this chromosome, which is one single chromatid, makes a copy of both the chromatids and start looking like this. You understand? So this only happens during the S phase. It is very short-lived state, but just by looking at the books and diagrams, we think that this is the chromosome which is present in cell all the time. It's not the chromosome which is present in the cell all the time. You understand how? And when you do this, you are actually keeping this is still 2 chromosomes, right? Chromosome 1 and 2. You understood? There are 2 chromosomes still. Either they were in chromatid form or they are in sister chromatid form. They are still, you can count it as 1 and 2 only, right? Maha?
Yes, sir.
There are 4 chromatids, 1, 2, 3, and 4, but they are together. But how many chromosomes are there? 2 only, right? Let's do it again. This is chromosome 1. This is how a chromosome is. And this is chromosome 2. How many chromosomes do you see?
2.
2.
How many chromatids do you see?
2.
2. Because in this phase, the chromosome is the chromatid. But when S phase comes, it replicates and makes the copies. Now let me ask you the question. How many chromosomes do you see?
4.
2.
2.
How?
1 and 2. Just 2 chromosomes. But how many chromatids do you see? 1, 2, 3, 4. You understand? This is how the chromosome number stays the same, but the DNA gets doubled. Now, all these chromosomes in this X shape, this X-shaped chromosome means it has doubled its DNA. Okay, Ma?
Yes, sir.
The X shape of the chromosome tells us that the DNA is doubled. S phase has been done. Now it needs to divide. So what happens during during metaphase? These chromosomes align at a plate, and from 2 sides, 2 spindles attached, and then take half, like 1 chromatid towards one side and this other chromatid towards other side. Do you understand now?
Yes.
Yep. That's how the ploidy remains the same. That's how the chromosome number remains the same. And also the DNA gets doubled and segregated. Clear, everyone?
Yes, sir.
Okay, so write down. There is no increase in the chromosome number. Okay, now about S phase also, let's write down some more things. Write down DNA replication. DNA replication, just write it as points. DNA replication takes place in the nucleus. Okay. DNA replication takes place in the nucleus. And centriole duplication. Centriole duplication. There is something called centriole. Now, let me go to Iram. Iram, centriole. Time to be the teacher. Last chapter.
Yes, sir.
What? Yes, she's happy. Tell me, what is a centriole?
It is a small hollow cylinder, non-membranous structure that occur in pair. They are only found in animal cell.
Yes, right. And what do they do?
The main function of centriole is they give rise to spindle fiber, which is
Exactly.
important for the cell to undergo division.
Exactly. So now pay attention here, everyone. Just to explain you, no one will ask you to draw this diagram. But to begin with, every cell is like this, right? Let's just say this is nucleus, this is cell. Inside the nucleus, you have chromosomes. Okay? Say yes or no?
Yes.
When the S phase comes, this chromosome, and along with this, there are centrioles. So as Iram said, they are cylindrical structures, which are like one centriole is like in this direction, then the other centriole is like in this direction, right? This is how they are present in the cell. Yes? So one nucleus and a centriole. Okay? Now what happens? Pay attention. S phase comes. What is happening? What I am doing here? What happened? Did something happen? Nothing happened. Ma, can you see all the, am I audible?
Yes, sir.
Yeah, okay. I thought I lost the connection. Can you see what I have done? Let me go
Yes.
back a little. Yes, it was like this to begin with. Then came S phase. And this is what it started to happen. What I'm doing here?
Did you cut them?
No. Don't just go on my action. I'm not cutting them. What I'm doing? These were chromosomes with one chromatid. Now they are chromosomes with two chromatids.
Oh,
So they are
yes.
replicating. Yeah. Understood what is happening? DNA is replicating in the nucleus. Also, this thing, what happened? Centriole replicated in the cytoplasm. Okay. Is it clear? This is what it happens.
Yes, sir.
Then in the next step, the nuclear membrane dissolves. There is no nuclear membrane. And in the next step, all the chromosomes start aligning on the metaphase plate. Understand? In the next step, these two pair of spindles start moving towards the poles. One to this pole and one to this pole. Okay. Then they start forming the spindles. Each spindle goes and bind with the. Do you understand what's happening here, everyone? Imagine it like a
Yes.
movie. This happens. This is metaphase, late metaphase. In the next step, you will see that for each the chromosomes, we see that each half is separated. Do you understand now what I'm doing? The spindles are getting shortened and they separated two chromatids. So it was like X, right? There were two chromatids together to give a X. Look at my fingers here. So one goes on one side and the other goes on the other side. Can you see this?
Sir, so where's the centromere?
Where's the centromere? So centromere is on the chromosome. So if I
So
show
they
this
get
one,
separated from the centromere.
yes. So if I show this one in zoomed detail, so this is one chromatid. This is the other chromatid. Yes. In the center, this is centromere, and on two sides there are two proteins called kinetochore, and the spindles are attached to these when they pull. Okay. So when they pull, right from the center, they divide. So every chromatid still has a centromere, but the centromere splits. Okay. And this one goes this side and this other one goes this side. Is it clear, Iram? This is what you are seeing here.
Yes, sir.
Yeah. Clear? Now, after some time, you will see that all of this has reached, all of this DNA has reached one pole, and this DNA has reached another pole. Okay. Now the spindle fibers are not required anymore, so they are either shortened or they are lost. And now it is a good time for the cell to start making a furrow. This is called a furrow. It starts invaginating, and then after some time, it becomes two different cells. Okay. You understand? It becomes two different cells. So if I remove this, magic. Two different cells are there. Now in the next step, these are not required now. And all this DNA gets concentrated and they are single strand, like single nucleus. And then the nuclear membrane starts to appear again. This is called telophase. Clear? From one cell, we made two cells. Yes? Clear to all?
Yes, sir.
Okay, so now let's write this whole process. Write down. DNA replicates in the nucleus, or DNA replication takes place in the nucleus during the S phase. DNA replication takes place in the nucleus during the S phase, and simultaneously centrioles duplicate in the cytoplasm. Okay. Simultaneously, centrioles duplicate in the cytoplasm. Is S phase clear to you?
Yes, sir.
Now let's come to G2. Write down G2 phase. Now G2 phase is another gap or growth phase. So in G2, write down. During the G2 phase, the cell growth continues. The cell growth continues and cell synthesizes some proteins required for division. So in G2, you can see that some synthesis is also happening, right? In G1, it was just the growth. Yes or no? Growth, but no synthesis. Like no DNA synthesis. But protein synthesis must be happening because to grow also you need to synthesize things. In G2 also, the cell continues to grow and it also starts forming now proteins which are required for division. Okay, because now after the G2 phase, it will commit into the M phase, the starting point. Can you see this, everyone? Yes, no?
Yes, sir.
Okay. Now write down. Next, let me delete this. A special case. Let's write down about G0. G0 phase. Some cells, some animal cells, do not exhibit division or do not undergo division. Some cells do not undergo division or only divides occasionally. Or only divides occasionally. So the question is, where are they spending their time when they are not dividing? Because if something is G1 phase, then automatically it will go into S from G1. From S, it will go into G2. And then from G2, it will go into M. There is no way out. So there is a special condition where they have to get out of this cycle, which is G0 phase. Write down. These cells, like the heart cells, I think Iram gave this example, right? Heart cell. I'm using the same.
Yes, sir.
These cells, yes, these cells, like the heart cells, enter an inactive stage. Oh, okay, before entering, write down. These cells exit the G1. So G0 always comes when, see, it is automatic that anything that has finished the M1 phase, it will go into the G1. So from G1, you have to exit. You have to make a voluntary exit. So these cells exit the G1 phase and enter and enter an inactive stage known as quiescent stage, G0. Now the condition here is that G0 sounds like, the cell is not doing anything. It is out, it has opted out. So, but the cell is living, right? Just to be living, you have to do a lot of activities. Remember class 10th, life, you studied a chapter called life processes. Was it in 10th or 11th? I don't know.
10th.
10th, life processes, right? So if any system is living, what are the characteristic features of it being living? What distinguishes living from dead? Remember, metabolism was one characteristic. If a cell has to be living, respiration, metabolism, consciousness, right? These three characters has to be there. So even though few cells enter into the G0 phase, quiescent stage, it does not mean that they are metabolically inactive. This was a neat question. Which of the following statement about G0 cells is incorrect? And one of the statement was that these cells enter a metabolically inactive stage. So what students often do is they know this inactive word is there. It is called an inactive stage. So the moment they read inactive, they think, oh yeah, it makes sense. But remember, if a cell goes metabolically inactive, it will not be living, right? Yes or no?
Yes, sir.
Yeah. So write down characteristics. Cells in this stage remain metabolically active. Cells in this stage remain metabolically active, but no longer proliferate or grow, but no longer grow and proliferate. Okay. So always remember this thing. Now there are two types of divisions. One is mitosis and the other is meiosis. Okay. Now talking about mitosis, mitosis happens in animal cells in almost all our cells, like all the body cells. There are two types of cells called somatic cells and germ cells. Anyone knows the meaning, the difference between somatic cells and germ cells? Yes, no, something? No? Okay, so write down.
So germ cells are sperm and ovum.
Exactly. Perfect. Was it Iram?
Yes, sir.
Yeah, right. Germ cells are the gamete cells, sperms, ovum. Okay, they're also known as sex cells, which gives rise to zygote upon fertilization. Somatic cells are every other cell of the body apart from germ cells, apart from the sperms and the ovum. So apart from reproductive cells, all other cells in the body of every organ is somatic cell. Is it clear, everyone? Yes?
Yes.
Yes.
So write down mitotic cell division in animals. Mitotic cell division in animals is seen or takes place in diploid somatic cells, in all somatic cells, basically, in all diploid somatic cells. Okay, so in animal cells, animals are mostly diploids. Let me tell you some things about biology now that you should remember. It will help you in class 11th, class 12th, and always, even if you go higher in research. There is something called ploidy. Before I move forward, these are some things. Let's take a detour from your syllabus now to give you some foundational concepts. So there is something called ploidy. Anyone knows what is ploidy? What does ploidy means? Write down. It is the copy number of genome. Okay. Ploidy is the copy number of genome. Every organism has a different number of chromosomes. How many chromosomes do humans have? Humans?
46.
46.
So ploidy can either be haploid, or it can be diploid. It can also be polyploid, like triploid or tetraploid. Now the word di means 2. Haploid is 1. So it has just one copy number. It has one set of the genome. Diploids have 2 set of the genomes. Polyploids have multiple set of the genome. If it is triploid, 3 sets, tetraploid, 4 sets, hexaploid, 6 sets. Is it clear? We denote haploid by n, 1 copy, diploid by 2n, and polyploid will be 3n or 4n, etc. Okay? Is it clear?
Yes, sir.
Animal bodies are mostly diploids. Most of the animals are diploid. Okay. Haploid are gametes. Our gametes are haploids. That's why a sperm is a haploid cell. Ovum is also haploid. So when sperm and ovum fuse together during the process of fertilization, then 2 haploid together give rise to a diploid zygote, from which our whole body is formed. Is it clear, everyone?
Yes.
Animals are not polyploid. We do not tolerate. Animals cannot tolerate polyploidy. Animals are mostly diploids. And write down, animals don't or do not tolerate polyploidy. So humans cannot be 3n or 4n, never. But polyploidy is very well tolerated by plants. Plants can tolerate polyploidy. Is it clear? Rice and wheat varieties that are grown in agriculture, they are polyploids. For sometimes tetraploid, sometimes hexaploid also. And you all have read about this. If not read, have you read about germination in plants? Germination, seed germination?
Yes.
No? Yes? Okay. In plants, there is a tissue called endosperm. You know about endosperm? It is present in the seed. Endosperm is a triploid tissue, 3n. and it is present in all the seed producing plants. So plants can tolerate polyploidy. In animals, only gametes can be haploid and their body has to be diploid. In plants, plants tolerate haploidy also. Plants can be haploid, plants can be diploid, and plants can be polyploid. I think now Maha would like to become plants even more because they can tolerate everything. They have no problem with any ploidy related issues. Animals, whenever there is any change in the ploidy, there are disorders that happens in the body. Ma is thinking, yeah, good decision it was to be plant, right, Ma?
Yes.
Yeah. Yes. Don't feel so bad about being an animal because being an animal also has a lot of perks. Yes. So you understood ploidy, everyone?
Yes.
Yeah. So mitosis happens in animals, right now. In animals, mitosis is observed. This is another question. So that's why I'm preparing you with explaining all the ploidic thing. In animals, mitosis is observed in diploid somatic cells. In animals, mitosis is observed in diploid somatic cell. Because apart from these cells, what other cells an animal body has? Haploid germ cells. Okay, and they do not show mitosis, they show meiosis. Currently, we don't have to worry about that. Is it clear, everyone?
Yes, sir.
Yes. But write down in plants, mitosis is observed in both haploid and diploid cells. In plants, this is the difference between an animal and a plant. Mitosis is observed in both haploid and diploid cells. Okay. Understood?
Yes, sir.
Now, before I go now to the most crucial stage, now before I start the marriage ceremony, this was all Haldi Mehndi Sangeet. Just go through everything till now in your notes and let me know if you have any doubt. Then I will start with the sub stages of the M phase. Take 2 minutes, go through all the notes and let me know if you have any doubt. Because now the actual ceremony is going to begin with chromosomes and everything.
So,
Yes.
so
Hira.
these all phases like G1, S phase, and G2, these all take equal number of time for synthesis or growth?
G1, G2, and S. We're talking about
Yeah.
the
Yeah.
interphase. So in the interphase, yes. So it's more or less, same only. It's just like it is difficult to distinguish between the three. In the S phase, we can only come to know when the DNA starts replicating. You can see whether a cell is in the S phase, whether the cell has not reached S phase or it has done the S phase. So if you see single chromatids, you will say that the cell has not yet doubled its DNA. That's why all the chromosomes are just single chromatids. If you start seeing this X-like appearance, you will know that S phase has been done, right? but basically the cell is just growing. So it is very difficult to just say that where did G1 stopped and S started. Do you understand? So that is
Yes.
one difficulty in actually figuring out the exact timing. But yes, more or less when it's grown, after that it replicates the DNA, and then it grows a little bit more with some protein synthesis required, and then it enters the cell division, M phase. Okay.
Yes.
Yeah. So the boundaries between G1, S, and G2, which you are asking, the answer to your question is it's not very well elaboratively. It's not very well elaborate, so you cannot, because once you go into the M phase, there are clear cut things that you can see and tell whether it is prophase, metaphase, anaphase, telophase. Cut to cut distinctions. That is not present in the different sub stages of the interphase. Okay,
Yes.
and the um, the distinction which is there, or the difference which is there, is very molecular. So it would not be the appropriate level to elaborate that thing to you. So just know that if you see the cell cycle, you have to just understand that interphase is the biggest, and it has G1, S, and G2, and their characteristic properties. That's all. Okay. Any other doubt, Maha and Aisha? Shall I start with the ceremony? Shall we start with the ceremony now? Okay. Maha, no doubts?
No, sir.
So let's start with the mitotic division. Mitotic cell division. This is the marriage ceremony. Now, write down. In mitosis, the ploidy of the parent cell and the daughter cell cells. And what is ploidy? The number of chromosome. Okay. So in bracket, I'm also writing number of chromosomes. So the ploidy of the parent cell and the daughter cell remains same. Therefore, or hence, mitosis is also known as equational division. What do we mean by equational? It remains equal. The number of chromosomes, what was in the parents, the same number of chromosomes will be there in the daughter cells after division. So that's called, so the number of the ploidy remains equal, so it's called equational division. Is it clear? Yes or no?
Yes, sir.
Now, this mitosis can be divided into, write down, mitosis can be divided into 4 stages. 4 stages of karyokinesis. Okay. And they are named as prophase, metaphase, anaphase, and telophase. So these are the stages of karyokinesis, division of the nucleus. If I go up, look here. So, sorry, I have marked it wrong. Should have written it. So these 4 stages are the stages of karyokinesis. And then one last is cytokinesis. And together, karyokinesis and cytokinesis, together, all of them are called M phase. You understand? So first the nucleus divide. And it has 4 stages, and then the cell divides, because for the cell to divide, all it has to do is to make a furrow and then restrict and form 2 cells, separate. So these 4 are the stages of the karyokinesis during mitosis. Is it clear?
Yes, sir.
Now, anyone of you has NCERT with you right now?
Yes, sir.
Yeah. Can anyone confirm that does your syllabus also has sub stages? Because prophase also has its sub stages in mitosis. So I think it's not in mitosis, right? Just check in the book and let me know. I think it's there in meiosis. Yeah, meiosis is little more convoluted, but I think for mitosis it's simple, no? Just the prophase,
Yes, sir.
metaphase, anaphase, telophase.
Mm.
Okay. Did you see the NCERT confirmed?
Yes, I've seen.
Okay, so let's start with prophase then. Let's see how the marriage initiates. Now, the nucleus was resting, and during the S phase, it has gone duplication. So the nucleus has double the amount of genetic material ready. All the chromosomes are in the X form ready. Okay. Now, what to do now? Prophase is the beginning, and what happens in this? Write down. The characteristics for every of these stages, we have to just know the characteristic features. So write down characteristic features of prophase. I'll use different color then. First thing that happens in prophase is that the chromosomal material, like the chromosome, when the nuclear membrane is intact, okay, so inside a nuclear membrane, when chromosomes feel like they are in home, and in the homely environment, we all, we do not carry ourselves in a very compact manner. It's like spread. Like if you see my room, everything is just present somewhere or the other. But if I have to go out all of a sudden, I'll start arranging things, right? I'll put my laptop in the sleeve and put it in the bag, arrange a bag, get dressed, get all my wallets, like and other accessories, and then I'll move out of the house. Yes. So I arranged everything before moving out. But when I'm living in my room, everything is not arranged. You understand? It's not compact. Yes.
Yes, sir.
So prophase is just the beginning of all the compaction that is going to happen so that all the chromosomes align at metaphase. So before that, what happens? Write down chromosomal material which was spread in the nucleus, nucleoplasm. So nucleus also has its own fluid called nucleoplasm, right, Maha?
Yes.
Yes. Chromosomal material spread in the nucleoplasm starts to condense, to form compact mitotic chromosome. Compact mitotic chromosomes. And what is a compact mitotic chromosome? This is exactly that I was drawing and telling you. So compact mitotic chromosome, write down. These chromosomes have 2 chromatids. You can understand why, right? Because it's replicated. Yes. These chromosomes have 2 chromatids. In the bracket, you can just write down sister chromatids attached together at the centromere that splits. Okay, Hiram? Is it clear? Do you understand? This is logical, right?
Yes.
The bride and the groom both are trying to compact things now because the day is near. So these chromosomes have 2 chromatids attached together at the centromere. And what else was happening in the cytoplasm during the S phase? So in the nucleus, DNA was duplicating. What was happening in the cytoplasm? That whole anime. Exactly. Centriole can I call it centrosomes? Because centrosomes have 2 centrioles, right? 2 centrioles make 1 centrosome. So this is 1 centriole. It's like a disc-like structure. And this is another centriole, another disc-like structures. And both of them are present together. Then together they are called centrosome. And separately, this is 1 centriole, this is 1 centriole, right? So write down second point. So this is about the nucleus. So what's happening at the house? What's happening at the marriage hall? There must be some arrangement started. So write down centrosome that duplicated during the S phase. Centrosome that got duplicated during the S phase in the cytoplasm. So here you have used the word nucleoplasm. Here you have to use in cytoplasm. Begins to move towards opposite poles. Remember? Yes or no?
Yes.
Towards opposite poles of the cell. So basically this is what they are saying. One reaches here, the other reaches here. And they start forming out these. Okay. And this structure here, which starts coming out, is called esters. And Iram, tell me, did I tell you that these esters or spindle fibers are made up of microtubules? So tell me what was microtubule? Remember? Microtubule. Yes. What was it? It was the cytoskeletal elements, right? Cytoskeletal element, people. Those elements which make the the structure of the cell. Microtubules, actin filaments, actin myosin, this word you must have heard. Actin filaments, intermediate filaments, microtubules. So these things which come out of the centrosome are called esters, and they are made up of microtubules. Okay. Write down the esters. The esters forms mitotic apparatus with spindle fibers. This ester, so ester plus spindle fibers is equal to mitotic apparatus. Mitotic apparatus is when they get attached to the chromosomes in the center, then it is called mitotic apparatus. Okay, this full-fledged, which starts pulling the two half DNA on one side and half DNA on other side. So just remember this one thing: ester plus spindle fibers is equal to mitotic apparatus. This was asked in NEET. By the way, we also have to do some NEET previous year's questions based on chapter 8, which is cell, the structural unit. Okay, so that we'll do in the next class. Is this clear? What happens in prophase? So the in prophase, the the preparations have begun in both. In the nucleoplasm, chromosomes have started compacting, and they are having 2 chromatids each. Okay, and in the cytoplasm, the centrosomes started moving towards the opposite pole and started giving out esters, which together with the spindle fibers will make the mitotic apparatus in the coming some time. Okay, so basically the preparation is happening at both the places, at home and at the mandap or at the marriage hall together, in the nucleus and in the cytoplasm. After some time, the nuclear membrane will dissolve because you no longer will be in the home. You will reach the place, you will come out in the cytoplasm. U means chromosomes, which are getting, so they first will get lined at the metaphase, and then they will get separated. So next is metaphase, and write down one more thing, one last point. At the end of prophase, prophase, the end of prophase. Remember, Iram and also Maha and Aisha, I told you, the endomembranous network of the cell is very, very vivid, right? Endoplasmic reticulum starts from the nuclear membrane and goes all the way to the cell membrane. Yes?
Yes.
Yes.
Yeah. So do you think if they are intact in the cell can divide? It will hold up the cell, right?
Yes.
Yes.
So before the cell starts arranging all the DNA at the metaphase, it has to dissolve all the membranes. So you will not see these membranes. So at the end of prophase, write down Golgi complex, because this is also membranous, ER, and the nuclear envelope and the nuclear membrane. Is not visible. Is not visible in the cell. It basically dissolves. We can form it again. We have to make it again after. But currently, the cell has to undergo a division, an event. So at the end of prophase, the cell becomes like a soup with no endomembranous network, no nuclear membrane, and the chromosomes now, these typical X chromosomes now are ready to come into the cytoplasm. You know, it's like the Dulha and the Dulhan coming to the what do you say, marriage hall. So they will all come together for the event, for the union, okay, at the metaphase. So is it clear till here? Any doubts? Prophase is clear?
So in
Yes.
prophase, the cell organelles disappear, right?
Yes. So majorly the endomembranous organelles. You don't have to, basically dissolve and disappear ribosomes and mitochondria, because anyways they are not forming a membranous network. The problem with membranous network is they will not allow the spindle fibers. So you understand what is the problem. See, if this is the cell, and if one centrosome is here, if one centrosome is here, normally there will be a ER network like this. There will be a Golgi network like this, right? So when they will give spindle out, it will just not reach anywhere, right? You understand? There is a problem. Too much crowded, too much of membranes, too much of compartments. Do you understand, Iram? So we have to get rid of all of this so that now these spindles can reach the center. Okay? Iram?
So are you able to hear me?
Yes, yes. Do you understand what I said? Could you hear me? No, I think Iram is not able to. Let me just wait for her. But this is where I will end today's class. Maha and Aisha, do you understand today's class, Maha?
Yes, sir.
Okay. So in the next class, we will take from metaphase. Remember, the ceremony has begun and preparations has started on both in the nucleoplasm and in the cytoplasm, and the endomembranous network has dissolved. Now, in the next episode, in the next class, we will bring both, like all the chromosomes at the metaphase. Okay, let's wait for the Iram. Yes, Iram.
Yes, sir. It's clear.
Yeah. Okay, is it clear? So I was just telling Aisha and Maha that we have reached till the beginning of the ceremony. In the next class, we'll start from metaphase, where the chromosomes will come at the plate, central plate, to just agree, to say Kubul hai. Okay, then see you in the next class. Take care and please revise this thing, and see you in the next class. Bye-bye.
Thank you, sir.
Bye-bye.